I am Dr. Monica from Noida Institute of Engineering and Technology. I am working as Associate Professor in Noida Institute of Engineering and Technology, which has been affiliated with Dr. APJ Abdul Kalam University, and I am taking the subject Pharmaceutical Microbiology having code BP 303T. This is my lecture number 5. I have already covered, if you have gone through my lecture number 4, then in the lecture number 4 I covered the nutrient media, how we can prepare nutrient media, how we can what are the various agents which we need to add into the nutrient medias. So that agents we learned, and we also learned the techniques, how we can prepare those stabs, how we can prepare the slants, how we can prepare the Petri plates containing nutrient media which can be further used for the isolation purpose. So that for that you need to follow the lecture number 4, that is what ingredients we need to add, how we can isolate, how we can that is make the slants and stabs and Petri plates for further use in the further experiments. So in this case, in this lecture I would like to focus on the isolation part, how we can isolate once we have prepared the media, once we have prepared the slants, once we have prepared the stabs, once we have prepared the Petri plates. After that, how we can isolate those cultures, how we can cultivate our pure cultures within the lab. So in this case, first of all I will like to focus on the 4 I's. As I already told, we have to, our whole microbiology is revolves around 4 I's. Now what are those 4 I's, and after that I will try to cover up the isolation techniques, various isolation techniques for pure cultures, how pure cultures can be isolated in the lab. There are various techniques we will cover. So first of all, 4 I's. In my first lecture itself I covered these 4 I's. Now what these 4 I's indicate? One is inoculation, second one is isolation, third one is incubation, and fourth one is your identification. Now what these 4 I's stands for? First is inoculation, means transfer process. Inoculation stands for this transfer process, means we are trying to transfer our that is species from one culture to the other. This is a process by which we can transfer using the sterile inoculating loops, sterile inoculating needles. We can transfer our cultures, we can transfer our microorganisms from the one culture, one media to the other media. So inoculating loops are always required for the inoculation part. Second one is isolation, means we can isolate our pure cultures, we will obtain our pure cultures from the mixed cultures, the cultures which contain lot of microorganisms, the samples of soil, the sample of water, the sample of saliva, the sample of air, they all contains number of microorganisms, lot of microorganisms are there within those samples. That samples are known as mixed cultures, in which there are lot of species, number of species are there. There will be Staphylococcus aureus, there will be E. coli species, there will be Salmonella species. So mixed cultures are the culture in which there are number of microorganisms are there. So for isolation we need techniques to isolate them into a single species. To take out the single species from the mixed culture, we require some techniques so that those pure cultures can be developed in the lab. So next one is incubator. Incubation is a technique in which we provide the environment in which microorganisms can grow. We have equipments, we have incubators in which we can place those microorganisms, those species, and they will grow, they will get an environment in which that is suitable for their growth. After that comes the identification part. Now how we can identify? So the whole microbiology revolves around these 4 I's: inoculation, transfer process, isolation, means we will come to know about the techniques to obtain the pure cultures. Incubation, we will be able to provide the environment in which those microorganisms can grow, and identification are the techniques by which we can identify those species, whatever pure cultures we have developed, whatever single species we have cultivated, we will be able to identify those species. And if we cover these 3 I's, that is inoculation, isolation and incubation, these 3 whole I's put together that is known as cultivation. Means if we inoculate, we isolate and we incubate and we develop a pure culture, we generate a pure culture, that is what we call as cultivation, that we have cultivated our microorganism, our species in the lab. So, cultivation includes the whole these 3 I's, one is inoculation, isolation and incubation. Once we have cultivated, after that after the cultivation part we can identify our species. So, where that my identification techniques we will come to know how we can analyze that whether we are able to cultivate the species we wanted or not. After that, this is inoculating loops. As I told, inoculation, for inoculation we need inoculating loops. Now, what are inoculating loops? Inoculating loops are the wires, or these are, as you can see in this diagram, these are the inoculating loops. They are having a loop which is used for transfer process, but before using it for transferring, or that is for transfer of microorganism from one sample, from one media to the other, we need to sterilize these loops. Whatever loops we are using, we have to sterilize those loops, and those loops are sterilized using the Bunsen flame. Flaming is the main technique by which we sterilize the loops, and they are heated till they become red hot. They will be put into the flame and they will remain in the flame for the time period till they become red hot. In this way they will get sterilized, and if loops contain infective proteinaceous material, then we need to add them, dip them into the chemical disinfectants, so that they become sterilized. Whatever microorganisms are there with on the loop, those microorganisms get killed, and we will be able to use these loops for further transfer processes. Next comes the isolation. Now, what is isolation? As inoculating loop I told is used for the inoculation part, for the transfer process, and similarly in case of isolation, if we want to isolate our pure cultures, means we are trying to isolate, obtain a single species from the mixed culture, means the culture which was containing number of microorganisms, now we want to isolate, we want to obtain pure cultures from those mixed cultures. So, how we can isolate, how we can that is single microbial species, how the single microbial species from the multiple culture can be isolated. There are various techniques for that. Now, what are those techniques? First is direct inoculation method. We can use direct inoculation in the media, and once we will directly inoculate in the media, the colonies will form and we can obtain the colonies. And second is streak plate method, third is spread plate method, pour plate method, serial dilution method, and micromanipulator methods. These techniques we will learn today about how we can isolate our microorganisms using these techniques. First of all, I would like to focus on the streak plate method. Now, what is streak plate method? As I told before, in each and every isolation process, what you need to make the first media. Until and unless you do not have the nutrient media, you will not be able to isolate any of the species, and media must be solid in nature. And so, that is why if you go through my lecture number 4, I have covered up that how we can prepare the nutrient agar medium, how we can prepare solid media in the lab. So, first and foremost requirement is to prepare sterile nutrient solid media, nutrient agar medium, which must be solid in nature. The first step, you need to sterilize the grassware. Second step, you need to make the nutrient media, that must be sterile. Autoclaving say you have to sterilize that media. After that, you will take that media to the aseptic room, where we can transfer those medias in the Petri plates. That in the Petri plates, in the liquid form we will transfer into the Petri plates, and in the Petri plates we will get the solid agar medium. Once we will keep it at room temperature, with that Petri plate which contains nutrient agar medium will solidify, and those solid media plates are further used for isolation purpose, for streaking techniques, for spreading method, for various methods for isolation in which we can do isolation part. So, first we need to make sterile Petri plates containing agar medium that must be solid in nature. Agar medium you have to add, and agar medium will get that must be solid in nature. So, when we keep that Petri plate in room temperature, that media will get solidified, and in this way you will see that in the first, when we try to isolate, we have to take the mixed culture. We must have first, we must have that is Petri plate containing solid media. Second, we must have the culture from which we need to isolate the pure samples or pure species. That culture is known as this, in the test tube we always have the mixed cultures. That mixed culture may be a water sample, tap water, that mixed culture may be a saliva sample, that mixed culture may be a blood sample, that may maybe a soil sample, maybe a air sample. So, mixed culture will contain number of microorganisms. So, we have to make a dilution of that mixed culture in the test tube. Once we have made get the mixed culture, from that mixed culture we can isolate now our pure species. Now, how we can isolate? There is a technique. First, the loop you have to take, that loop must be sterile. As I told, by flaming process you need to sterilize the loop. So, using flame we have to sterilize the loop. Second, loop is inoculated in the test tube. That loop must, we will put that inoculating loop into the culture media which contains mixed number of microorganisms. That may be water sample, I told, that may be air sample from where that prepare that culture media, we have mixed culture media. We can prepare from the air sample, we can prepare from the soil, or even we get from the that is market itself, which contains number of microorganisms, that is known as mixed culture, which is having number of microorganisms. We will put that inoculating loop in that culture, and after that, once we have put the we took the inoculating that is culture, that drop will come into the inoculating loop, and that loop will then first be put on the plate, and on the plate we will try to make streaks over the plate. The plate which was containing solid media, we will take the loop and we will try to streak it out. First of all, we need to divide that Petri plate into various sections. Now, various sections are, we can make them as 1, 2, 3, 4. In this 4 section, first what we will do, we will streak that loop which was containing, that is number of microorganisms or mixed culture it was taking, and that loop we will move like this. Now, what we are doing, we are trying to spread the culture, that culture which was containing number of microorganisms. By this technique we are trying to spread that culture into the plate, and after that, once we have streaked out in that culture in the first column, then again what we will do, in the fourth step we will again sterilize our loop. We will let it cool, so that when we will move further, that must not be, that is contain any of the microorganisms, that must not get contaminated with the air. So, that is why we sterilize, and we will let it cool for some time. Again we will place that loop here, and we will again take that sample and we will try to move like this in the second part. Similarly, when we want to move towards fourth or third column, then also what we need again, we have to sterilize the loop. Whenever we are moving from 1 to 2, from 1 to 3, from 3 to 4, at each and every step you have to sterilize, you have to cool that sample. Whatever samples you are taking, you have to cool those samples like this. These you have to put like this, 1, 2, 3, 4. And what you need to do, you have to move it like this, and whenever you are going to move from 1 to 2, you have to sterilize the loop, you have to cool the loop, and after then again you have to place like this, and you can do the streaking within the Petri plate like this. So, there are various types of streaking, there may be parallel streakings, there may be multiple streakings. So, streaking, what streaking will do? Streaking will spread the multiple cultures, the culture which was having number of microorganisms, those cultures will be spread with the help of, that is, streaking technique, and once you will spread, the microorganisms get isolate. Here you will have the number of microorganisms when you will put the loop, but as it is moving from one part to the others, from other to the next, and from next to the next, here we will have a colony, we will get a colony which will be having a single microorganisms, that will not have number of microorganisms. The mixed culture was there. From when we take a loop full of mixed culture, we will move, we will try to spread on the Petri plate, and once we will spread that, this type of colonies you will obtain. As you can see in this picture, these colonies are very much concentrated. In the first part colonies were very much concentrated, but as we were moving from first to second, second to third, we can see the concentration is going on decreasing. The number of microorganisms which was there within the first area number 1, that was highly concentrated, area number 2 was less concentrated, and area number 3 you can see is very much less concentrated. Number of microorganisms will be decreased to maximum extent, and you can now see there are isolated colonies in this Petri plate. Now those isolated colonies you can pick, those colonies are the that is contains these type of colonies will contain single species of microorganisms. We will pick that colony, we will put that and these colonies will only develop once we have placed our, that is Petri plates containing streaking, on which we have already done the streaking. We will place those Petri plates in the incubator, and in incubator they will get the condition in which the microorganisms grow, and at there the environment, they will get the environment for the growth, and we will see that these type of colonies will get generated after that is 7 to 14 days. We will be able to see those colonies will be visible, which were invisible in the starting point. So these colonies will be visible after 7 to 8 days, and we will see in the first column there will be the colonies will be highly concentrated, in the second column colonies will be less, and in the third column we will have much more less colonies or much more less number of microorganisms, and in the fourth case if we say we will get that is isolated colonies will be there in the fourth part, and those isolated colonies will contain single species of microorganism, and that species, that colony, when we will pick and we will add it into the fresh media, then a new culture will generate new if we that colony if we put in a fresh media, that colony will contain single type of species, single type of microorganism. So each and every colony which is being isolated like this, that those colonies always contain that is single species. In this way, by streaking plate we can obtain the pure colonies. Similarly, main aim is to spread. You have to spread the mixed cultures, because mixed culture contain number of microorganisms. Until and unless we have not spread that culture, we will not be able to obtain the colonies. After that comes spread plate. What we can do in spread plate, as I told, you have to, that is main parameters are same, that is you need to make a sterile media, you need to have a mixed culture to isolate. So these two things you have to make ready, that is first is Petri plate containing media, and second one is your culture media. Now what is the difference between spread plate and pour plate? As in case of streaking, we were doing the streaking to spread the microorganisms, we are doing like this streaking, that pure mixed culture was going to spread, and in the end we will get the pure that is isolated colonies. So those isolated colonies will generate a pure cultures. Similarly, what we will do in the spread plate, in spread plate we will, that is like this, this is a Petri plate which contains agar medium, that agar medium must be sterile, there must be no microorganisms within that media. After that you will take the inoculating loop, this is the inoculating loop, and with the help of inoculating loop we will add 1 drop of that is mixed culture, and once you have added 1 drop of mixed culture, what will happen? This mixed culture will spread. We will try to spread with the help of L-shaped glass rod, like this swirling is done to spread this drop. Ultimately we need to spread the culture, whatever mixed culture we have added to the Petri plate, sterile Petri plate containing agar media, we have to spread that. And how we can spread? We can spread with the help of that is L-shaped glass rod. So once we have spread by swirling, sample is spread evenly over the surface of agar using sterile glass spreader, L-shaped spreader is swirled and to spread the drop of a mixed culture, and once we have spread, we will incubate in the incubator and we will provide the conditions in which microorganisms can grow. After that incubation, after 7 to 14 days, what we will see? We will observe the colonies like this, is you can see colonies will develop. Typical spread plate results means colonies, isolated colonies you can see in the plate. These isolated colonies contains single type of microorganism, single species, those are taken, those pure, that is colonies are isolated, picked up and added to the fresh media, and when we will add these single colonies to the fresh media, they will generate pure cultures, they will contain single type of species, single type of microorganisms. Similarly, this is spread plate, we have covered, that is streaking technique. Second, I told you about the spread plate technique. Next come the pour plate. Now, what is the difference between spread plate, that is streak plate, and pour plate? Streak plate, in streak plate we do the streaking for isolation, in spread plate we spread the culture, and in pour plate what we are going to do, we have to take the sterile Petri plate. First we will take a plate, sterile Petri plate, and that Petri plate will contain nothing, that must be sterile, there must be no media within the Petri plate. We will add first the, that is species, that is mixed culture, we will drop a drop of mixed culture within the Petri plate which is containing no media. Once we have added the drop of that culture, mixed culture within the Petri plate, we will spread the whole, that is this this colony, this culture, mixed culture, we will spread within the Petri plate, and once it will get spread within the Petri plate, what you are going to do next, you have to add the media. Now, what is the difference between spread plate and pour plate? In spread plate what we did, we added our drop on the solid media, media was there, but in pour plate drop is added before adding the media. So, drop of mixed culture is added, that culture is spread over the Petri plate, after that we will add the media, agar medium which must be solid, that must be liquid in nature, so that we can pour that media on the culture. So, once we have poured, we will let it cool, that is that it becomes solidify, after that swirl it when it is liquid containing, that is mixed media, mixed culture, and that is agar medium is there, we will swirl the whole plate, so that each and every media and each and every colony gets spread, each and every microorganism will spread within the Petri plate, within the whole media it will spread, and by swirling we will mix it, and after that we will incubate it in the incubator, we will solidify and we will place it into the incubator, and in incubator what will happen, we will be able to generate, that is we will provide the conditions in which the microorganisms can go, and once the microorganism will go, you will see the colonies like this. Now, what is the main difference between spread and pour plate is, in spread plate what we will see the colonies will develop on the surface of the, that is Petri plate, but what we will observe in case of, that is in case of pour plate, the colonies will get generate deep into the cultures, not on the only on the surface, we will see my colonies will develop on the surface as well as the colonies will develop on the, that is deep inside the culture media. So, that is the isolation technique, that is a pour plate technique, in which we will get the colonies even deep into the culture, the colonies will grow and we can isolate those colonies, and those colonies, isolated colonies are added to the fresh media, and we will get, that is what we call as that is pure cultures. We add them into fresh cultures, these colonies, and we will grow those colonies. These colonies will generate pure cultures, that is those colonies, those that is samples now will contain single type of species. This is what we learned, that is in, that is first we covered the, that is streaking technique, streaking technique we were doing the streaking. In spread plate we have spreaded our culture on the Petri plate, that is using the media was there, then we added the culture, mixed culture, we swirled and we spread the culture and incubated, and after incubation we got the colonies, pure containing single species, single type of microorganism. In pour plate what we did, we added first, that is sample, that is mixed culture, after that we added the media, and after that we swirled and completely spread the, that is culture within the media, and after that we incubated, and after incubation we got the colonies deep into the culture as well as on the surface. In this surface colonies will develop, in spread plate also surface colonies will develop and in streak plate also we will get the colonies on the surface and not into the deep culture. So, this is what the next one is serial dilution. Now, what is serial dilution? See, if we go for serial dilution, in serial dilution what you will do, you will use the nutrient growth. In previous slides I have shown streak plate, spread plate and pore plate. For that we need actually the solid media, but in this case for serial dilution means we are going to dilute our samples, and when we are going to dilute, we are reducing the number of microorganisms within each test tube. So, how we can do the serial dilution to isolate our microorganisms or bacteria? This is the serial dilution, that is first of all, if you see these test tubes, in these test tubes what we are going to add, we will add 9 ml of media. The media we can add water, distilled water we can add, dilution, that is solvent, dilution factor, dilution media we need, that must be sterile, there should be no microorganism within that media. So, this all test tubes like 1, 2, 3, 4, 5 test tubes are there. In all the 5 test tubes what you need to do, you need to add the media, that nutrient growth we need to add, that will do the dilution of the microbial culture. So, first of all, media must be sterile, all test tube will contain 9 ml of, that is nutrient media, nutrient growth which must be liquid in nature, and after that what you are going to do, you will add 1 ml of original mixed culture, which contains number of microorganisms, we will add 1 ml of that culture into the first test tube. Now, how much dilution we have done? We have diluted our sample 1 ml sample to 10 ml. That means 9 ml was already there, we added 1 ml and we got 1:1:10 dilution, means we have diluted our culture 10 times. Further, next moving forward, we will shake whole test tube containing 1 ml mixed culture media and 9 ml growth. We will shake it, and what we will do, we will get after that we will transfer 1 ml to the next test tube, we will get 1:100 dilution. After that we will again shake it, again take 1 ml from the previous test tube and add to the next test tube, and we will get 1:1000 dilution, means we have diluted our original sample 1000 times. In this case we have diluted our sample 10,000 times. So, as we are going forward, we are moving forward, we are diluting our samples, means we are decreasing the number of microorganisms within each test tube, and we will get like this. If we inoculate or incubate this sample, we will get like Petri plate like this, that colonies will be highly concentrated. If we incubate those samples containing only 1:10 dilution, then they will contain number of microorganisms. But if we incubate this sample, put them into the Petri plate, then it will just generate colonies which will be highly less, number of colonies will decrease as we move from here to here. When we move from first Petri plate to last Petri plate, we will get reduced number of colonies. Similarly, there is one method what is known as micromanipulator. This is a specialized industrial based technique in which single microorganism is isolated with the help of a micropipette. We will be able to see those single species in the microscope, and those single species we can pick with the help of these micro capillaries. Those single species are picked, and then those single species are added to the fresh cultures, and we will be able to generate whatever species we want to pick. We will see the image, and those species will be picked with the help of micropipette, and then we will transfer it into the fresh culture, and we will be able to obtain the pure culture of microorganisms containing single species. So, these are the references which you can follow for this, that is, you can follow Prescott, you can follow Pelzar, you can follow Chiaraneja. These are all the references which you can follow for these all practicals or all these techniques by which we can isolate our mixed culture, we can isolate our pure cultures from the mixed cultures. Thank you so much.